So let's talk now about the action potential. Remember we said we have the graded potentials, and these usually do not reach the threshold. If that change reaches the threshold, and we said the threshold is like a critical level, the minimum amount of change in the membrane potential to trigger an action potential, then we will have an action potential. And action potential follows a rule that is saying all or none. So either we have action potential or not have action potential. Once the change, if this is the threshold, once the change reaches the threshold, then we will have action potential. If the change did not reach the threshold, then there is no action potential. If the change barely reached the threshold, like here, then we will have an action potential. And action potentials will not differ. Okay, so whether it barely reached the threshold, okay, or it significantly reached the threshold, we will always have very identical action potential. So action potentials are very identical regardless of the amount of the stimulus causing them. So all of these ABs are identical, same speed and intensity, and they do not decay over time or distance. So once we have it, that's it. It's like a wave that is not going to stop till it reaches the end of the axon. This is what does it mean, all or none. For the actual changes in the action potential in the nervous system, they are a little bit different than the one we saw in the muscles. In the muscles, we said we did not talk about any threshold in the muscles. This wasn't existed, okay, or it did not exist. We did not talk about it, okay. So this is one difference, that there is the threshold. Other than that, it's pretty much the same concept for depolarizing. Usually with depolarizing, there is sodium gates open, sodium ions gates open, sodium rushes in. This would reach to a more positive value. At this stage, sodium gates close, potassium gates open, and potassium leaves the neuron. And this will cause repolarization. So pretty much the same in here and here, very much similar or very similar to the one we saw in the muscles. However, when this repolarization value drops down, actually with the muscles we saw that it would flatten out and it reaches the same level immediately. But in the nervous system, no, it goes even below the original membrane potential. It goes below the original membrane potential value. This is something called hyperpolarization. Hyperpolarization. So when the neuron is restoring its membrane potential, it even go beyond the resting state into an area called hyperpolarization. And in a short explanation of why do we have hyperpolarization, this is a result of the slow closing of potassium ions gates. Sodium ions gates will open fast and close fast. Potassium ions will open fast but close very slowly. Like think of a rusted gate that it's giving like the sound, something like this. That are responsible for hyperpolarization. But this is an important thing happening because it allows action potential to travel in one direction, which we are going to see how. Action potential. So action potential would start as we saw in the muscles. Sodium ions would rush into inside. Okay, this is inside. And they will cause changes in the membrane potential into this area. This will cause more sodium ions to open, and they are going to diffuse, okay, and open the following sodium ions gates. And this will keep continue, like dynamo effect. So we already talked about this. This is very similar to the muscles. We already talked about this. Now, the difference is that, as I said earlier, that action potential is going only in one way, from the cell body, this is the cell body, toward the axon terminal. These are axon terminal. So action potential traveling only one way. We don't have an action potential that goes backward. This does not happen. So we need to understand why action potential is always traveling into this direction, from the cell body toward the axon terminal. And that's what we are going to deal with now. I will do some erasing here, and let's draw it in a different way. So this is the membrane potential. This is the plasma membrane. And here, what I'm going to do, I will draw like lines to make sections of the axon. So this is part of the axon, okay? Maybe another part here, or another part here. In these parts, all of them, they have sodium gates and they will have potassium gates. Always we have sodium gates and we have potassium gates. And these both are voltage gated channels. So what happens is that when sodium comes in, it will diffuse. Okay? When sodium comes in, it will change the charge here and it will diffuse. Sodium here will change the charge here. This will cause the gates to open and we will have more sodium coming in and diffusing in both directions. So sodium will diffuse into here. It will open this gate and the new sodium will diffuse and open this gate. Remember, this is happening through time. This is a timeline. And this is by milliseconds. Each segment will take around 5 milliseconds. When sodium comes in and it changes the membrane potential, what is going to happen is that the potassium channels, since there is this change, they will open and potassium will start to leave. Okay? Yes, there is a delay between sodium gates open and potassium gates open. There is a delay. This delay would be enough for the sodium to diffuse to the second section and open the gate. Okay? Once this is happening, or as this is happening, as the sodium are diffusing, potassium gates will open and potassium would start to leave the cell. And this later would restore the membrane potential, meaning that this is going to close. The same situation here. Once sodium is diffusing in and it's diffusing to the second section, potassium gates will open and potassium would start to diffuse out. And it will repeat itself. Again, because of the changes of the membrane potential, potassium gates will open and potassium will start to diffuse out. The same thing, because here we saw a change, then these potassium gates will open and diffuse out. So far, this is what's happening in action potential, very similar to the thing how the action potential is spread in the muscles. But in order to understand, yet we did not understand why this is happening only in one direction. So in order to understand this, we have to draw the actual action potential lines, the depolarization and repolarization for each one of these segments. So when sodium ions came in, they are causing depolarization, right? So as they are coming in, they are causing depolarization. And they are diffusing out to the second section. But this and this are not happening at the same time. There is difference in time. So this is happening, okay? And after a while, after 5 milliseconds, this would start to happen. But during this 5 milliseconds, we will have repolarization. So while this is going up, this is going down at the same time. And it will repeat itself. Here, this is going up, and now we are in the third section. Now this is going down while this was going up. I can't draw them both at the same time. Okay, I can't use this. So that's, you have to imagine them, that the line going down in repolarization and the line going up in depolarization is happening are so you have to imagine that the line going down in repolarization here and the line going up here in depolarization happening at the same time. Okay, now because of this and because of hyperpolarization, the neuron is not able to receive a new stimulus, or this portion of the neuron is not able to receive a new stimulus. To understand this, let's put it on a separate graph. So we said we have depolarization, repolarization, and then let's use green, and hyperpolarization. Now, if a stimulus arrives here, or arrives here, or arrives here, it's very unlikely, almost impossible, to have another action potential. This will not happen. Also here, if the stimulus arrives, another stimulus arrives here, we will not have a return back of the action potential. No, it will keep running, it will keep going down. Okay, the same thing here. If the stimulus arrived while the that segment of the neuron is in hyperpolarization, we will not have another action potential unless this is like really extremely strong. Remember the stairs? Okay, so here in the hyperpolarization, we need more stimulus to reach the threshold. So here we need more stimulus again to get it back to the threshold, which is not going to happen. These sodium ions diffusing back, they are not strong enough to cause a new action potential. But here, while this is at the resting state, they can easily induce an action potential. Like see, for example, here, this sodium, when it's diffused into this area, the membrane potential is at rest, and it's easily can be increased to pass the threshold. So it's easier always to get an action potential in the forward segment, not in the backward segment. And that's what causes the action potential to go only in one direction. Again, because of these sodium ions, when they try to diffuse back, the membrane potential will be in the hyperpolarization area. So we cannot induce another action potential. You can think of it, it's already in action potential. You can't have a double action potential. You can't have a double action potential. It's exactly if you are sitting and someone telling you sit down, but I'm already sitting. Or if you are eating and someone tells you like eat. Or if you are driving and someone tells you like drive. Okay, I'm already driving. I can't do double driving. Okay, so this is the same concept. We can't have double action potentials. There is one, it has to finish before we start a new one. So this is why we are always going to have action potential moving in one direction. Let's see now the difference between the myelinated and non-myelinated nerves or axons in form of how the action potential is transmitted. So for the myelinated or for the non-myelinated, you can think that we have sodium and potassium channels all over the way, all over the axon. So the action potential has to jump from here to here, from here to here, from here to here, and so on. This is time-consuming process. Like this is like 5, 10, 15, 20, 25 milliseconds. This is non-myelinated. But if it was myelinated, then there is the myelin sheath, there is a myelin sheath, or extended, and there is a myelin sheath. Okay, so in case of the myelin sheath, the sodium and potassium gates are only in the nodes of frame var. So action potential has to jump like longer way, which will make it faster. So here, from here to here, like this is 5 milliseconds, here this is 10 milliseconds. And we traveled much further distance comparing to non-myelinated. So this is the case of the myelinated axon. And you can see this on this graph. Okay, so here, for example, if you are looking at both are starting at the same time. The signal is starting, the blue line is starting at the same time. But this is traveling much faster. This is traveling much faster. So it will end, it's at the axon terminal while this is still here, like in the first third. So this is the difference between myelinated and non-myelinated axons in how fast they can transmit the signal. For how the following neuron is receiving the signal, this could be that following neurons or the postsynaptic neurons could be excitatory or inhibitory. Okay, so either it's going to excite, the neuron, causing it, like for example in the muscles, to contract, or to inhibit it, causing them to relax. So this will depend on several factors, but one of the factors that it's depending on the types of the neurotransmitters, which will take us to talk about the synapses. This is a pretty straightforward concept, so I'm going to go through it very quickly. That whenever two connections, or whether there are two neurons or neuron with another structure meet, that they are going to form a synapse. We have chemical synapses that would require a chemical signal, usually a neurotransmitter, and then there is electrical synapse that is basically controlled by voltage gated channels. When there is depolarization in one, the other one will sense it and the signal will be transmitted. For the neurotransmitter systems, we have several systems. The most important one is something called Kolodziejczak system. This is based on acetylcholine. Okay, and we have two types of receptors that are receiving acetylcholine. The first type is narcotic receptors. These are in the neuromuscular junctions. Okay, and the other type is something called muscarinic receptors. Usually these are in the smooth muscles and even in the heart. Okay, these are muscarinic receptors. Then we have different other classes like amino acids. Some scientists consider them also a class of neurotransmitters. We have something called biogenic amine. These are groups of several amino acids, small number of amino acids can act as a. There is an additional class called neuropeptides. These are several amino acids connected by peptide bonds. So all of them are variations of the amino acid class. The main thing I want you to understand from this, that the effect of these neurotransmitters, whether it's going to be excitatory or inhibitory, is depending on the receptor in the postsynaptic cell. In the cell that is post the synapse, this is the synapse. Okay, so here we have the receptors. So these receptors would determine the effect of the neurotransmitter. So the receptors would detect the effect of the neurotransmitter. So it means that we could have acetylcholine acting in two different receptors. One receptor would cause the cell to be excited, the other cell would cause the cell to be inhibited. So the effect of the neurotransmitter is depending on the receptor. So the effect of the neurotransmitter is dependent on the receptor that the neurotransmitter is binding to. Binding to that receptor can cause either depolarizing, in this case it will be excitatory, or hyperpolarizing, which in this case will be inhibitory. Okay? Think of this as like a music or food. These are the neurotransmitters. Some people would like them, certain types of music or certain type of food. Other don't like them, don't like the certain type of food or music. But it is the same music, the same food. Both of them are tasting the same type of food. Some people will like it, some people will not like it. Why? It's different receptors. This is exactly the same concept for the neurotransmitters, and their action is dependent on the receptor. So that's it for this chapter. Let me know your questions, and we can have more discussion about these concepts.